NGS and bioinformatics. I hope you all enjoyed a hearty breakfast and had a chance to take in the view, of course. I mean, what could be better than Mumbai rains, cloud-covered hills, and the calming waterfall just nearby, right? Minus, yeah, 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 minus the red alert, of course. Now that we are refreshed and ready, let's dive into another exciting and enriching day ahead. The past 2 days have been filled with amazing learning and collaboration, and today promises just as rewarding. To kick off things, we have a highly informative session on basics of Linux and an overview of ACTREC pipelines. This is a foundational topic, especially crucial for those of us working with bioinformatics tools and genomic data. Without further ado, it's my pleasure to invite Dr. Vishram to take us through this session. He will guide us through Linux, its significance in bioinformatics, and the pipelines used at ACTREC.
Hello, am I audible? Hi, so good morning to everyone. Today I'll talk to you about two topics. First one is basics of Linux, and the second is about the pipelines which we are using at in our department. Right? So we'll start with basics of Linux. This is the outline of my talk. I'll tell you what Linux is. Right? I'll tell you why you should care about it, how the files are organized. How do you install programs? I think this was a query which came up yesterday. Is it required for bioinformatics, or can you do away with it? And some useful commands to take you through. So let us start with understanding what is Linux. So Linux is a generic name for a family of operating systems. It is not one operating system. For a family of operating systems which are based on something called Linux kernel. Right? So kernel, and this is actually a patent, a trademark actually. So Linux is a trademark owned by this guy Linus Torvalds, because he created the Linux kernel. I'll tell you what kernel is in a while. And it actually started as his hobby project, right? And he wanted to create something free for everybody to use. One of the known operating systems that time which was used was Unix. This was even before, sometimes during when Microsoft was even starting. So they wanted something free. So he created this Linux kernel as a hobby, right? And Linux kernel on itself is not much useful. You need something called utilities also. Utilities are the ones which you interact with actually, right? So they released the first operating system, or the basic operating system, in 1991. And GNU is actually provided by another group or a guy who owns that group, Richard Stallman. And it provides a lot of programs. One of the most important programs it provides is Shell. And this guy, Penguin, this is the Linux mascot. His name is Tux. And once you see this guy, you should know that people are going to talk about Linux. So Linux, I said, is free. Free as in it is an open source software. And you will now be confused, like why should anyone give their programs or software for free? And it actually allows other programmers to develop their own software and to release them without any licensing fees attached, right? So this helps them, the other people, to modify it, incorporate it in their own system without having to pay for it. Does it have any advantage, or just a gimmick? It has advantages. It increases the reliability and transparency. That means a lot of people can test your code, look into your software, and fix any problems, or fix any problems which might be present, right? So it is actually an excellent tool to promote your company's or institution's image or a product, and it allows other people to build upon what you have already done. So it gives new abilities and promotes innovation, right? And Linux is one of the most famous examples of open source softwares, right? So here is an image which you can see here is which explains you what kernel does and how the operating system is organized. So Linux kernel sits here. It actually talks directly to the computer hardware, whichever computer you are using, and on top of that is this group of utilities called GNU system utilities, which usually people interact with. And so this two actually made the first operating system. So once you have it, it is not much useful, because not everybody is accustomed to be using terminals or command line interfaces. You need something called window management software. This actually is the interface which you see when you open a computer. So these are called desktop environments or graphical desktops, and on top of this is your application software. Application software can be anything. For example, Chrome. Chrome is the application software which you access when you open a window or a Windows PC, or a similar similar window management software for Linux is KDE or Genome. So these are the names of those graphical environments. Now these are those two people who actually developed this operating system. This is Linus Torvalds. He gave us Linux kernel, and he also gave us something called Git. We will talk about Git. It is a version control software, and GitHub is the online version of it, where you can use Git. And today, almost all softwares and programs are stored on GitHub, right? And this is another guy, Richard Stallman. He gave us GNU utilities. One of the famous utilities which you will be using and which some of you have used is the shell. And these two guys gave us this functional operating system which we are using. I mean the Linux operating system, right? So why should anybody care about Linux? Like, do you think that is it important to know about it, or is it just some fancy operating system which came out of someone's hobby project? Actually, you should, because Android softwares which are running on our smartphones, they are based on Linux kernel, and smartphones which use Android, they make up 43% of all smartphones in the world, right? So that means Linux does command a sizable market. Most of the internet servers are based on Linux. That means you are getting the internet on your computers or on your smartphones because there are Linux servers. And top 500 fastest supercomputers are actually using Linux. So I mean, without any exception, right? So that means Linux is important, but why don't you know about it, or why don't we use it on day-to-day life? In day-to-day life, the thing is, it is not widely used on desktop computers. It is a special. It has a specialized use, but these days it is gaining popularity. So is it suitable for daily use? The answer is based on what you would like to do. So it is okay if you just want to use web browsers. If you want to look at videos, music, images, and so on and so forth. One great advantage actually of Linux is that it has limited viruses and malware threats. It is free and open for everyone. It is used by almost all software developers and data scientists. So if you fall in this category, definitely Linux is for you. But it is not suitable for for the kind of for people who mostly work on Microsoft Office or similar programs, because Linux doesn't have an equivalent there. It has it, but it's not as good. So and the second is gaming. Gaming is a big industry. If you are involved in gaming, mostly Linux is not for you. So operating systems in Linux, they are called distributions or distros for short form, and there are two major types. Ones which are developed by community. That means a community of developers, software engineers who actually make the operating system and give it for free. So you can download this on your respective computers and use. Examples are this Ubuntu and Fedora. And second is enterprise based, which are actually made by certain companies. For example, Red Hat Enterprise Linux, right? So these are paid. So some people try to avoid it. But these days, due to the rising popularity of Linux, what has happened is that even Windows has developed software which says which can mimic Linux. So it is called Windows Subsystem for Linux, and you can download it for free on your computers, just like any other software, right? So coming to the business end, we are going to use this program called Shell. Shell is a very important program in Linux. It is an interactive program. That means you can interact with it. And how do you interact? You interact using command prompt, right? And to interact with the shell, you need to give a set of commands. You give in some input to perform tasks like copy files, compute values, and rename files, and so on and so forth. And this group of commands, when put into a file and executed as a program, makes a shell script, which which I'll show you in a while. So what do you see when you open a Linux computer or a Linux-based operating system? You see something like this. So this is a graphical window which you see. It is really basic. It doesn't have much. It only has this menu bar and some applications on the left-hand side. So although it is pretty much evolving now that there are different types of graphical systems. These are genomes, KDE, and Unity. Uh, and on this left hand side you will mostly see applications, right? And the most important application which we use is this thing called command prompt or command line interface. You just go to applications and there will be something called terminal, right? So this is how the terminal looks like. Most of you might have tried using MobiXterm today for the tutorial. So this line is called command prompt, right? And this ends with a dollar sign. Dollar sign is important. It tells you that that you can start entering your commands here. Once you see the cursor back, you can enter a new command. So this dollar, before that dollar sign, there is this user ID followed by an @ followed by the name of the computer on which you are logged in. So this gives you some information that you are logged in as a user in a computer, and you can now start giving commands. So this interface is called command line interface, and there are different kinds of shells which you can use. Dollar stands for a shell which is very specific called bash, right? So since shells are important, we will give some we'll just talk about shells for some time. So bash is the default shell for most of the Linux distributions, and it is used for writing scripts and managing processes. So there are a variety of shells available, just because it is so important to interact with the computer. That bash stands for bash born again shell. It is a GUI utility, as I said, and it is replacement for some previous shell. But there are others which include TCSH, Corn, and ZSH. So these are very specialized shells. So you may not be using anytime, but ZSH is a very advanced shell feature which incorporates all of this. So how do you find out what kind of shell you are using on your computer? It is really simple. To identify the shell, you type this command echo $shell, and once you press enter, you should see output something like this /bin/bash. That means bash is the shell which you are using, right? So this was for maybe Linux or some Windows based computers using MobiXterm. But if you are using a Mac operating system, which I am sure some of you have, when you say this echo $shell, the output is something different. It says bin/zsh because it uses a different shell by default. And you see this dollar prompt has changed. Instead of dollar, now there is a percentage sign. It changes based on the shell type which you are using. Now, to understand Linux, you need to understand the structure in which files are stored or the programs are stored in the system. So all files and directories, unlike Windows, they are stored in a virtual directory. It's not a real directory or a real folder, but the base of this directory is something called root. It is denoted by a / and everything in a Linux virtual directory is stored somewhere inside root, somewhere. So for example, if you download and install certain programs, they are stored in /bin. Bin stands for binary, that means programs. /etc for system files. /home is a very special destination. It is for home directory of the users. So when you log in, most probably you are in /home, right? So this is the comparison between Windows and Linux. When you have a Windows computer and when you try to look for your path, you see the first letter you see is C, followed by colon, followed by / followed by backward slashes, right? And then whatever the location of your folder. But when you do the same thing in Linux using a command called pwd stands for print working directory. It will tell you where you are. It says /home /wtp, whatever. So you see the difference is that Windows uses backward slashes, Linux uses forward slashes to denote its path. That being said, so all your programs, when you install anything on Windows, they are stored in program files, right? C/program files or whatever you have. But in Windows it is in /bin. See, bash is in bin. So these are the major differences between how a Windows operating system is versus how a Linux operating system is. And you can use a command called cd to traverse the directories. And the other important command which you would like to know is print working directory to find out your current location, right? So now that you know that there are directories, we should also know one important thing about Linux. All Linux operating systems are multi-users. That means they are made for a lot of people to be used at the same time, right? That means it can give rise to security issues, no? Because files from a user A can be used by a user B, right? And that is not desirable. So to to avoid that, and in general to provide security, why people say Linux is secure, it is because you can provide for permissions to your objects. Objects can be anything: files, your folders, your links, whatever is present in your account, right? And there are three major types of permissions for a given file or a folder. That means whether that file can be read, whether that file can be modified, or whether that file can be executed as a program. So these are called read, write, execute permissions. So this is one set of permissions. Second set of permissions is for the group of people who can use it. First is the owner, the user himself who made the file, whether he can use it or not, or what he can do with these files. A group is a set of a group of users. Like since I said it is a multi-user system, few users can be combined into a group, right? And anybody else. So anyone else who logs in as a guest, right? For example, what will they have access to? So these three permissions versus three groups gives you total a combination of 9, right? When you enter a command like ls-l in your home directory, maybe you see an output something like this. For this file, suppose my program is the file. The first three permissions tell you what the owner can do. Owner can read, write, and execute. The second three permissions tell you what the group of owners can do. They can also read, write, and execute. And the third tells you what the guest maybe coming to the system can do. He can just read and execute. He doesn't need to read. He doesn't need to write. Right? So this is what makes Linux really secure, and this is what it is actually known for. Right? So now coming to why should you use Linux for bioinformatics? Is it required, or can you do away with Windows? So the answer is that Windows is not as suited for bioinformatics as Linux is. It is open source. Linux is open source, flexible, and cost efficient. Cost efficient as in it is practically free. And most of the operating system, most of the bioinformatics tools which you are going to use are made for command line interface. So they are not made for a graphical window based operation. And the next most important thing which you should know about Linux is that it can handle large data sets very efficiently. When you are dealing with genomics data, the output is really huge in terms of GBs and maybe in terabytes also. So that those kind of files need to be handled very carefully, and that only Linux can do, or a Unix based PC can do. And then there are limitations to what graphical interfaces can do, because there are limitations to how many options you can provide when you are using a software, right? But that is not the case with command line interfaces. And Windows command prompt is also something which can take in commands and give you output. And these days they have also introduced something called PowerShell, which I think some of you know, that they can take inputs just like your Linux computer does in Windows. But they are paid softwares, so you have to buy Windows, right? Only then you can get the Windows command prompt, and they are not as versatile. Definitely because of this reason, they are not as used also. You have the terminal option with Mac, but Mac operating systems do not. The software for the Mac OS is not available as a separate software. You have to get the hardware, and the software comes along with it. So definitely these two are out of question, and that leaves us with Linux. Hence the choice of Linux for bioinformatics, right? Now that you know that we are going to use Linux, what do you do with it? Of course, most of you know that we are here to understand bioinformatics pipelines, right? Analysis. And this analysis which you do, or these workflows which you make, they need to be reproducible, efficient, and scalable, right? At the same time. And how do you do that? You have to use something called workflow engines, and they execute your programs in a chained input and output manner. That means you can take the input from one software or one tool and give it to other. For example, you can trim a file and give it to a pad and assembly, something like that. So, and that is done by workflow engines. So where do you get this? And these are, of course, all of them are open source and free. One of them is Bash. Bash, as I said, is the shell programming language, and it is very easy to learn. But it is not scalable, and it cannot run parallel tasks at the same time. That means that for small scale analysis, Bash is suitable. You can use it. Then there is something called SnakeMake. It is Python based. It is really easy to learn again, but it is not scalable. It is not as scalable. So it has its use, but it is limited. But it is still better than Bash. And the next and the latest one which we use and which we also recommend is Nextflow. So Nextflow is slightly difficult to learn, right? It is not easy to use also, but it does the job. It is scalable and it can run lot of parallel processes at the same time. That means you can run lot of samples, you can analyze lot of samples at the same time. So this is the workflow engine which some people were asking for. So you can use Nextflow. And of course there are others, there are always others, bpipe, make and cromwell. These are used under certain conditions, right? So when you make changes to your software or when you make a pipeline, you always need to modify it or change it, right? And what do you do with those changes? You need to track those changes, or in general, if you make any changes to a file, you need to trace them back, right? Once in a while, right, to recall, maybe some specific details. And large projects like some pipelines or whatever you are working on, sometimes you need to revert back certain files or the entire project. You need to compare the changes, like what changes you made to the software before or maybe after certain modification. And you need to identify who made the changes also. So how do you do this? You need something called a version control program. And here are these two major categories of the version control programs. One is centralized and other is distributed. And I'll not go into the details of this, but I'll tell you this. One of the famous version control softwares is Git. This again is provided by the same guy, Linus Torvalds, and it was given away for free. So people can use it, and it is used by almost everyone who has ever done any kind of coding, any kind of scripting, or made any kind of file. Okay. So and this is freely available on GitHub. So you guys can make your own accounts on GitHub and use Git and start using Git. So we will talk about GitHub in a while, right? So now that you know that you have to use Linux, you also need to know how to use it and where to use it. Mostly analysis is done on Linux servers. These are for faster processing, because they have large memory and they can do parallel computation. Also as you can see, a Linux server can connect to lot of PCs, and they can be anything. You can use a Windows based computer, you can use a Mac based computer or a Linux based computer to connect to a Linux server, right? You just need to obtain a username, a host name and a password from your system admin. I mean the person who is actually running this server, right? And Mac OS is also something Unix based. So both Linux and Mac can use this kind of command to actually log into a server. You can say SSH, your username and the password, and then you are in the server, like in the server as in you are accessing the server. So but Windows is a special case, and most of us are using Windows. So for Windows using PC to connect to a Linux server, you need to use special softwares like Putty, Cygwin and MobaXterm. Of this, MobaXterm is quite largely used these days, and it is available for free, and it provides you a terminal to connect. It only gives you an interface to connect. That's all it does, right? So now that you have a Linux server to connect to and a user ID, what do? You start with bash scripting, right? And here is an example of a bash script. Bash script, this kind of scripts or any kind of scripts are created using a text editor. One of the noted text editors are Notepad. Some people know about VI, that's fine. So this is how the shell script looks like. The first line you see here, it has a hash followed by an exclamation mark, right? So this is very special. The first line of a script is always very special. It tells you what you are using. I'm using here user/bin/bash. That is, this is the shell which I am going to use. That means all my commands which I am going to add should be from this bash. Followed by another hash. This is a comment. It tells you what this script is going to do. Followed by the commands, right? Whatever commands or whatever you want the script to do, you add it here. Commands are executed sequentially. That means the first command to execute it will be ls followed by who am I, right? And not only you make a script, you have to make it an executable. That means you have to tell the software or tell the program computer that it is a software, it is a script. So you use chmod plus x source.sh to actually make this script executable. Now it has permissions to be executed, right? And you execute the script using ./source.sh. That's it. So yesterday after the workshop, some people were confused as to how do you install softwares on a Linux system, and this is the part which is really tricky. For installing softwares on Linux systems, there are separate softwares which are called package managers. So these are graphical tools which take in your input as to what you want to install, and they will install it, upgrade it, or remove it, right? They connect to the online repositories and detect the dependencies, I mean whatever softwares are required for your program to be installed, and then they give it to you. So one of the famous installation package managers is called APT, Advanced Package Tool. It is command line based, and how do you do it? You have to use this command sudo apt get install and the package name. Suppose you want Trimomatic, you said apt get install Trimomatic, right? But this was quite rudimentary. So of late what people use is Conda. So Conda is a command line tool and package manager and environment management software for all operating systems, Windows, Mac and Linux. So if you want to install something, you can just say Conda install Trimomatic. And most of the bioinformatic tools these days are available through Conda. So you don't have to do any hard work. You just have to go to the tool which you want. They will specify the command, Conda command, which can be used, and you just install it on your terminal. And you type that command on your terminal, and it gets installed. There are older package managers like Yum, Pacman, and DPKG. Once in a while, what happens is you will not get these softwares on your package management tools or on Conda. What you have to do then, you have to install it from source, and this is the difficult part which Linux is infamous for. You download the source package, that means you download the actual code, you have to read the instructions what are written in that code, and then you have to make the executable. This is difficult, but I mean this is the least followed path, but when nothing works you have to do this. So some of the problems which are frequently encountered by people when using Linux are, first is about command usage. How do you know how that command is going to work, right? For example, there is a command ls. How do I know what to do with it, right? So in that case, you say man ls. Man stands for manual pages. That means these are instructions already encoded in the software, which will tell you which will give you a lot of information about how this command should be used. Second is software not found. Software not found is a very well-known error, and for that you have to verify the installation. Again, you can use that apt-get which I told you in the slide before to actually install the software. Then once in a while your system slows down, right? So to do that, to overcome that, you have to identify the program which is stopping it. You have to use this command called top. It gives you the a list of programs which are slowing down your system, and then you terminate them using a command called kill, right? Then this is the one which I told you gives Linux a lot of security, but it also gives you a lot of problems. It's called permission denied. That means you do not have the authority to use a file. How do you do that, and how do you overcome this? First you identify the problem, ls-l, this will give you the permissions, and then you change the permissions if you are allowed to change the permissions. And then something there is, if not, file not found. This is a very common problem which you might have seen on Google Chrome also. Like when you try to search something, it says file not found. How do you do? So how do you overcome this? There is a command called find. It is so common an error that people made a command just to find a file. It's called find, and that will give you give you the location of the file in your entire Linux directories. So I'll leave you with some basic Linux commands. First is the list to show a list of files. Second is cd, which says change directories. pwd to print working directories. cp to copy files. Move to mv to move or rename files. rm to remove files. And we have kept a cheat sheet. That means a set of commands which are routinely used on our GitHub page. So you can go to GitHub, mol himat and workshop 24, and you can find a cheat sheet there which will help you to go through any Linux system. So to summarize this talk, what I have told you today is that Linux is a versatile operating system. It is suited for bioinformatics. In order to use it efficiently, you need to understand its structure. And not only you need to understand how Linux works, if you are going to use bioinformatics, you need to use workflow automation tools, which will give you reproducible results, scalable analysis, and resource management. And bash scripting is a simple yet a powerful method to parse your output. So with that, I'll end first part of my talk. I'll be happy to take any questions. Yes.
Question, but maybe you can tell them what kind of computers are required to do that. So, like a typical panel-based, software, let's say 50 gene or 100 gene panel, or an RNA sequencing targeted panel. What kind of computing requirements are there? So that is something that people, I'm sure, won't know about, especially who have not done this kind of thing, and whether they can do all this on a laptop. So you can tell them.
So, okay, I'll tell you what you can do on a laptop. You can analyze really small files on your laptop. Like small files are like something which has like, some 200,000 reads. If you have a FASTQ file like that, you can do it on your laptop. But of course, you are not going to analyze this kind of data. When you have large data, as you said, like, something for gene fusions or for large panels or for a decent sized panel, maybe of 100 something genes also, you need you need a server which has some at least around 200 threads. Some huge RAM is required. For example, you need to have 500 GB of RAM. No, sorry, 500 GB of RAM, and storage is required. So you have to use the data, you have to store the data. So even to access the data, you need like really huge storage. For example, like 15-20 TB of storage is required. And after you have analyzed this data, maybe on your server, you need to store it somewhere. So you have to also make provisions for that. Like I think there are requirements where you have to store, keep the FASTQ files for some number of years. So either you can use cloud-based storage, or you can have your own institutions storage. But that needs to be really huge. Huge as in like terabytes huge. Yeah.
Vishram sir, I have a query that for the Linux part, you are using a term that's scalable. It's kind of very scalable. And when you are talking about the Linux versus Python, then you are talking that it's not the scalable. So what thing actually this scalable things?
So,
because
yeah.
people will
I
gradually...
didn't, okay, I'll go to that part. So I was not talking about Linux being scalable. I was talking about this being scalable, right? Yeah. Scalable means how many samples you can analyze at the same time, right? And how huge can be your process. For example, we'll see some pipelines today. They are like really huge, and bash scripting is definitely not suited for those. And with suppose you have 100 samples to be analyzed at the same time, this is not something which you will like to use. Like bash is not something which you will like to use. So SnakeMake is actually a Python-based workflow engine. It is not Python. It is a workflow engine, and it is not as scalable as Nextflow, but it is decently scalable. That means you have to make modifications to it to make it scalable. That means it can on its own do suppose 50 samples, but if you want to analyze 100 samples at the same time, you need to modify it, which is not always desirable. But this guy here, it is slightly difficult to deal with him, but the kind of results it will give you are amazing, like nothing sort of amazing, and very few people actually try to use it. Dr. Nikhil, I think, was the first person who I saw using it, right? And yeah, that was great. This even like really big companies avoid this, but it is amazingly good at scaling and doing a lot of stuff at the same time. That's parallelization.
I'll explain to you what this means. So in pre-Vishram era, like when I started, we didn't have bioinformatician. I think Shweta and Gaurav were there at that time. So how to do this? So I was very familiar with bash, and we used to do bash scripting pipelines. All pipelines were made in bash, and that time Nextflow was very, very new. So if you have to, let's say you do, and you're running a service, so you have to maintain TAT, you have to maintain all of that. So you get 15 samples in a run. The way a bash pipeline will work is that it will take one sample from start to end, one sample from start to end. When that gets over, it takes the next one. And that is a problem, because somewhere along the line it gets a hiccup. Let us say somebody else accesses the server and runs something, and there is for even a microsecond a temporary resource overload, or some error comes up. It will abort the pipeline. Next day when you come, pipeline has been stopped at first sample, so everything goes back by one day. So that is where bash sucks. Nextflow will take all the samples together at once, and to the best of my knowledge, run everything in parallel. So everything gets, everything undergoes BWA, everything undergoes SAM tools, and takes it step by step. Let us say light went. Okay, real life scenario. Light went, server light went, backup went, everything went to hell. Armageddon. So next day you come, you'll find your pipeline stopped there. You can resume it exactly at the place where it stopped. So you don't have to go all the way again. It will tell you where it stopped. You can dig up the work directory. You can find out what error was and all, and fix it. So those sort of things, it's very good. Last thing, again, I'm picking your brain, Vishram. Please tell them, suppose I am new person. I don't have, it takes 40 lakhs odd for a server, 30 to 40 lakhs. It's expensive. Plus, it is not just server liya, usko, you have to keep it like a precious wife. You have to keep it in the humidity-free area. You have to put glass enclosure, all that sort of thing. We are in institute. All these things are there because IT already does all that. So we have to beg them, dalne dena, that sort of thing. But I don't have any of that. Now I want to use cloud computing. So can you tell what, we have used that. So pros and cons of cloud and how to go about it.
So, okay. So what he's saying is, you want to avoid buying a computer. You want to use someone else's computer. So cloud is actually someone else's computer. It is given by Amazon. So Amazon will give you, whatever it will give you a username, a host, and you are the Windows, this user, and you connect it to their server, right? And you use it. But the thing is, it is really costly. That's first thing. Second is that it involves a lot of intricacies when you actually deal with, doing parallelization there. It will work as a normal PC. So, but you have to do, if you have to do parallelization, I will say there is nothing like, using Nextflow. So even with Amazon Web Services, paying a lot for it, they don't provide you, they don't allow you to run a lot of samples at the same time. It will run one by one on a really, just a decent computer, and it will give you slow output. So for both time, energy and money, I would say cloud computing is not the choice you should go for. It is good if you don't have any other option. But if you have, you should invest in, your own server, or at least your IT department can go and buy you some server and you can use it. So that definitely is advantageous.
How about Linux workstations? Can we use that for some mid-level works?
Mid-level works, exactly. It is suited only for small analysis, I would say. But for, something like transcriptome, if you want to do on a workstation, it will not do actually. It will be really slow. For even on big servers, transcriptomes are slow. So for normal workstations, it will take maybe days, right? Or in a whole genome, if you are already doing whole genome, it can take a week for really big, even for 10 samples.
So this whole field is set to blow apart. I personally feel that we are at a crucial juncture here, where everything is written in this old school kind of code and software. Okay, what the world is revolutionizing right now, why Nvidia is $1 trillion company, is the use of AI to do super kind of gigaflop, teraflop calculations, and that if it hits bioinformatics, it will really accelerate all processes, all pipelines. Even with our server, which is very expensive, lot of things, it takes one day, I think, for a transcriptome analysis, which is ridiculous. Aur kya chahiye tumko? It's just that the computational processes are inefficient. But the downside of that is everything has to be rewritten to make use of those GPU processes. So That is something I don't know why somebody has not addressed. Someone should.
Well, maybe NVIDIA is actually trying to. I think they have some goals, yeah, but they will take money. Yeah. So I'll give you a short overview of what pipelines we are using at ACTREC. So bioinformatics, as you know, applies informatics to informatics, which is maybe computer science, maths, to large-scale biological data. In our case, it is genomics, right? And bioinformatic tools, when or algorithms, when executed sequentially to analyze this data, is what we call as a pipeline. And this pipeline should be accurate, reproducible, and they should have QC at different steps to ensure robustness and traceability, right? So here is an example of a pipeline. What it is doing is that it is taking two inputs, sample sheet and the directory, and it is throwing out one output, FASTQ. In the during the process, it is giving out some intermediate files. That's fine. So this is simple, like two inputs, one output. But pipelines can be really complicated. Look at this. It takes one input, uses a plethora of tools, and throws a lot of output, and then you have to maybe check for what you want. So with this kind of range, what do you use? Like, how do you define a pipeline for your purpose? So at our department, we have our custom-made pipelines to suit our requirements, and we use open source tools. That means we don't pay for it. On Linux operating system like Ubuntu OS, right? And the output of these bioinformatics tools is formatted or parsed using Bash and Python. These are again very simple and easy to learn. And these individual steps are executed in a chained workflow using Nextflow, right? And this allows us to have a scalable and reproducible scientific workflow or pipeline. And this Nextflow is written such that it can support most of the scripting languages. That means Bash and Python, right? So how to design a pipeline, and the design of pipelines is actually based on the experimental design, right? And we are going to talk about 5 types of pipelines here, which are actually 5 types of sequencing library preparation methods, right? Hybrid capture, amplicon, gene fusion, MRD detection, and whole exome. I'll give you an overview of all of this. So amplicon-based workflows use amplification, PCR amplification, to obtain target enrichment, right? And we use multiplex PCR to get uniform amplification efficiency. And what do we use it for? We use it to detect kinase domain mutation analysis in CML. And this is suitable for regions with high genetic variation, and because it has a lot of coverage. That means it has a lot of reads. So what is it not suited for? It is not suited for high GC and repetitive regions, and it cannot detect large indels or chromosomal rearrangements. And to analyze this kind of data, what do you need? So the output, first of all, is a FASTQ file. That means it looks like this. Most of you have seen, some of you will see today. So you have to go from a FASTQ file like this to an output like this in a tabular format, which most of you are accustomed to see. So how do you actually go? That's what a pipeline does. It takes you from here to here, right? And this is a simplistic workflow for analyzing the amplicon-based assay. And first step is, of course, you obtain the FASTQ. You trim it. You trim the adapters. That means you remove the adapters. You remove poor quality bases. This increases the downstream efficiency. Second step is to assemble the reads. Most of us, for most of our sequencing assays, we perform paired end sequencing, right? That means the read is sequenced from both ends. So we need to combine those, right? And that is done using a tool called Flash. It merges the paired end reads. It does some errors correction for the overlapping part. It updates the quality score, and then these longer reads are now good to go for mapping, right? We use BWA to assign genomic coordinates to these reads, right? That means they are mapped to the reference genome, and the reference genome which we use is 100 HG90. So once we this gives us a BAM file, and this BAM file can then be used to calculate coverage, perform variant calling, and other steps, right? So from this BAM file, we first calculate coverage, and the typical coverages which we see for amplicon-based workflow is approximately 10,000x, right? And we use Mutex to Wardict and Lofric as our variant callers to call variants, and this gives us a VCF file. And these variants are then annotated using NOR. what it does is it integrates information from various databases into a biological context to give the biological context to these variants. Right? So, but most of the sequencing methods which are used to scale to analyze large set of genes actually use hybrid capture based assays, right? So the first step in this assay actually is to design the capture probes to identify by identifying the target region which you want. And once you design the probes, you need to the regions of interest are captured by hybridization to the probes which you have designed, which have white-in labels, right? And this method has better performance than amplicon-based method with respect to sequencing complexity and uniformity, right? We use it to detect SNVs, indels, and CNVs in acute leukemia with a panel of 1300 probes for 137 genes. And the amount of captured DNA in this method is actually proportional to the DNA present in the sample, and that is useful in CNV detection using the read depth approach. And there is only one downside: it has longer hands-on and longer turnaround time as compared to amplicon-based method.
Sorry to interrupt.
Yeah.
Can you please explain that amount of captured DNA?
Sorry, this thing.
That.
See, you are going to capture capture the region which of your interest using this biotin probes, right? So whatever you capture is proportional to what was or present in the sample. If there is lot of it present, or if suppose there are 2 copies present, you will get 2. You will capture twice, right? And that means when you do CNV kind of detection, you will actually be able to see it because you are looking for read depth, right? So, so pipelines for analyzing hybrid capture based assays output are slightly involved. They are not as simple as you saw for the amplicon based data. You get a FASTQ from the sequencing machine, and we actually avoid the paired end assembly, and we directly obtain a BAM file from the Nexic machine itself. But this can also be obtained using customized steps. We proceed to variant calling using a battery of variant callers, and to detect indels we have 2 other tools, right? So once that part is done, we calculate coverage, and the typical coverages which we see for this assay is around 800x, right? We also do CNV detection using 2 tools. This assay is designed such that the probes which we are using, they are evenly distributed throughout the genome, which helps us in detecting copy number loss of heterozygosity using mosaic chromosomal alteration. And the variants which are obtained here are then annotated using NOR, NOTSV, or NCAVA. And one of the important things which you need to keep in mind when you are looking for the output from this assay is a metric called off-target capture, which determines the efficiency of the experimental step. Right. So we'll now move on to understanding how to use SMIPS based probes for ultra-sensitive detection of AML MRD. This is the SMIPS probe. It stands for single molecular inversion probe, and it has UMIs at both the ends. There is a target region. The target region is captured as in it becomes part of the probe. After some processing steps, it is sequenced, right? And the sequence read is such that there are UMIs at both the ends. These red regions which you are seeing, these are 4 base pair UMIs at both the ends. These UMIs are used for for eliminating random sequencing errors. What they do is after it is sequenced, these all reads coming from a single genomic fragment, they are aligned. They carry the same UMI and help the tools to identify a consensus from the reads which contain that UMI, right? So we use it to detect SNVs and indels in a panel using a panel of 156 probes for which has a WAF lower limit of 0.2%, right? So analyzing the data from SMIPS based assays is much different from what we saw earlier. Going from FASTQ till obtaining the consensus reads is actually done by a special tool called FGBio. It is a command line tool for analyzing genomic data. It provides its own best practices pipeline to obtain these consensus reads, and followed by once the consensus reads are obtained, we align it to the reference genome, and the BAM file is then used for coverage calculation, which is typically 1000x, and variant calling. So variant calling is done using 4 variant callers. One of them actually uses contextual error modeling for improved variant detection. These variants are then annotated using NOR, and the results are then analyzed further. So the next method, this is a homegrown assay in the lab. It is called Narasimha. We use it to detect chimeric gene fusions. It takes in RNA as an input, gets the cDNA, and after some processing, you add a special construct called SPOOM to both the ends of the of this cDNA, and this construct is then used for target enrichment, followed by sequencing this library. So one important thing to note about this assay, which is the uniqueness of this, is that it requires information about only one partner in the fusion, and it can potentially detect any fusion gene linked to that partner, right? And how do you analyze this? So this one is special in the sense that transcripts with or without any fusions, they are actually non-contiguous, and they are short reads, right? So this can increase errors when you are mapping to the reference genome. For example, if this is a transcript or if this is fusion, whichever you like to consider, if one part of it actually aligns to one part of the genome or one chromosome, and other aligns to some other chromosome, then it is an issue. Like, how does a normal alignment tool do this, right? So this requires use of special aligners. One of them is STAR. It stands for Splice Transcripts Alignment to Reference Genome, and in the absence of these aligners, we have high mapping error rates. That means which can give rise to incorrect calls, and it can take really long time, even with these aligners, to analyze this kind of data, right? So we use a customized pipeline obtained from NF-core RNA fusion, which is available freely. So this is the original pipeline. It takes in FASTQ as input. It takes like 5 fusion callers to give you one output, and we take this output and add a few more steps to it, right? So this pipeline allows for detection and visualization of fusion genes. We use this output to calculate coverage, and to standardize, and the output is standardized to remove false positives using a tool called MetaFusion, right? And the last assay I'll talk about is whole exome sequencing. This is done using a hybrid capture based commercial kit. It has some 2.2 million probes for 20,000 genes, and it can provide. It is said to provide 99% base coverage at greater than 20x sequencing. So it says that it is supposed to be sensitive to germline SNVs, indels, and CNVs at high coverage. High is like greater than 20x. But as compared to whole genome sequencing, it requires lesser sequencing. It gives smaller output, and it can be analyzed faster as compared to WGS. So the analysis of whole exome data can be done using this pipeline, and it is mostly a textbook example of how a pipeline looks like. So BAM file generation is done again using custom steps of onboard server. We use these GATK best practices to perform indel realignment, base recalibration, and to remove PCR duplicates. Once that is done, coverage calculation is performed. So here you have to calculate 2 types of coverages, the depth and the breadth, and the expected depth is somewhere greater than 20x, right? And we use variant callers like freebase, deep variant, and these variant callers need to be special in that they should be able to detect germline variants, right? And these variants are then annotated using NOR. So with that, I would like to end my talk, and I will be happy to take any questions.
For the CNV analysis, let's say like for the CNV analysis, let's say like some samples due to some kind of error in the sequencing, or we don't control the sequencer, like you do the library and then you send it out. So instead of 1 GB data, like you get somewhere around a 2 GB data or 1.5 GB data. So during that time, can we do subsample?
So we'll have full talk on CNV.
Okay.
On the mode of how we do it.
Thank you.
one choice always is like whether we should go for a Python based learning or a R based learning. So from that, what is your perspective from that question?
Python based learning is better, I think.
If you know Python, you'll know, end up knowing R. It's very, very similar in syntax.
Yeah.
That's the thing.
It is simple compared to R.
I have a query that for every step, like for the adapter tuning, alignment, for the variant calling, every step has so many tools kind of. You can use the X numbers of softwares and different software, different advantage. Some may be the kind of errorless, some may be the faster and everything. But at the end of the data, so apart from the coverage, should we take into consideration that we should change the software, change this step, or should we go for with the other softwares or anything?
So first is you have to look at your own data, how it looks like. If you know that there are, the sequencing quality is really good, then really stringent tools like Trigonometric need not be used. Second is that you can always compare them. There are a lot of papers which only do this. They just compare different tools and tell you what is efficient, what is fast, and what is good for daily use. So that definitely will help you. Second is that after each step, you should be able to see whether you have achieved what you wanted to. For example, if I wanted to assemble the pad and reads, the output of the reads, the output of that step should be, you should be able to look at it and say that it has done its job or it has not done its job. So before you actually standardize a pipeline, you have to do this. Like you have to look at each step and see whether the output is as you expected it is, as you expect it to
have
be.
Little play around with the steps.
Not little, actually you have to play a lot. In the lab, thankfully, the people who came before me have done this. So I need not do, but once in a while we need to. It takes time. It is a really time-taking endeavor.
Thank you, Dr. Vishram, for your insightful session. Now let's move on to our next session, that is Introduction to NGS File Formats. I invite Mr. Vaibhav to the stage, who will guide us through common NGS file formats, including the FASTQ, SAM, BAM, and BED files.
Okay, good morning everyone. वो laser. Good morning everyone. My name is Vaibhav and my talk is on common file formats in NGS data analysis. So these are the common file formats I'll be going over. So before all this, what happens inside the sequencer before we get the FASTQ? So as Swapnali ma'am and Shweta ma'am explained in their talks that our sequencing is done by sequencing by synthesis. So our template strand is like this, and then fluorescent oligos come and bind one by one. So when they come and bind, the control software of the sequencer takes pictures, and these pictures are stored in TIFF formats, and these TIFF formats are really huge, so they are not directly accessible. So the control software of the sequencer itself extracts the intensity from that TIFF formats and makes the CIF files. And at the end, we basically get BCL files. Those are base calling files. It has all the base calls and plus the quality. But we don't usually access BCL files as well. We directly get the FASTQs. because BCL files are basically everything pooled together, no? So we have to first demultiplex those BCL files to get the FASTQs of individual samples. So this is done. This can be done by two ways. Either the sequencer does it itself, or we can do it on command line using BCL to FASTQ command. So at the end, we get FASTQs for individual samples. So this is the general FASTQ file you can see, and these are 4 lines. First line is the sequence header. It has information about the flow cell ID and everything, like the unique instrument name, the run ID, flow cell lane, tile number, everything. We don't generally need it, but it's just there. Then this is the member of the pair. If it's the first or second read, then this is the index by which we demultiplexed. So these are the 4 lines. First is header, second is the main line. It is the sequence of our read. It is generally not generally, it is 151 bases long if we provide 151 cycles. That means whatever the number of cycles we are providing, it is that long. Third is the separator. It is nothing, just separates everything. And the fourth is Fred quality score. So I'll go over Fred quality score in detail in next slide. This is FASTQ generated by some long read sequences. This is the FASTQ from Nanopore. It's similar only, it just has different sequence header information, and the sequence is generally very long instead of just 151 or something. And its quality encoding is a little different from the normal Fred quality scores. Okay, Fred quality scores. What is Fred quality scores? So Fred quality score is basically Q is equals to minus 10 into log 10 of P. So it is the probability of incorrect base call. So if our base call if the Fred quality score is 10, that means one base call in 10 base calls might be wrong. So the base call accuracy becomes 90. So we can't deal with those files. We generally need Fred quality score above 30. That means we can tolerate one mistake in 1000 base calls. So our base call accuracy needs to be above 99.9%. So what are these CCCC? So these are basically quality scores, but they are not scored like 1, 2, 3, 4 because it is like very space consuming. So what they do is, this is the ASCII table. So these are the ASCII characters for individual numbers. So this C means, here you can see the it means the quality is 34 for that C. If it had been like G, then the quality would have been like 38, like that. So it basically consumes less space. So it is generally it is like efficient.
How we determine the quality? Like 1,100 how we determine it for each base?
The sequencer itself determines it and gives us quality.
Has these algorithms built into it. How it exactly does that, I don't know, but these are built into the the base calling thing, which already happens on board.
You have to blindly believe that. Because
So it has some probabilities. No, everything is maths and probability. Like based on this intensity, it will judge it by every cluster. Like this cluster's intensity met that or not. Was it less or not? If less, there is a statistical probability that this is good or bad. So it makes an estimate and gives a score.
because we are taking many decisions because of that. Because I am telling that the error is becoming higher, like I got your code. But in
So iron Terrant and Oxford PacBio, Oxford Nanopore, PacBio, all of these sequencers, they make the error because bases are called, when they are called consecutively, what 2-3 things can happen. One is that if a base is called consecutively, that means a homopolymer stretch is there, A, T, C, whatever. Your thing can get saturated with the intensity, and that is why these errors occur.
So that error is because many of the homopolymers, if they have a non-continuous bases like A, T, T, C and all, then
it is not a problem. It is generally not a problem. Single bases, even ONT is very reliable now, they say. Even this is very reliable. But if you see iron torrent, but if you see the indels is where it makes a call, is a problem. So you just have to look at a BAM file of an ONT or one of these. You will be appalled. Kya chal raha hai? That way.
So is this the quality of the overall output? That
Every base.
Single
Every
base.
base. That C, C, C,
You can
C,
see.
C. So those 150 lines are correspond to every base and its quality.
what is that, and what is the acceptable one? At what level?
So the quality, that depends on quality metrics. When you get the sequencing run, you will have a Q score more than 90%. So that is like a median score of 90% for Q30, right? That's what you are looking for. So Q30 more than 98% or something is very good. Whereas if you typically talk of ONT, it is Q20 they talk about. Now it is improving with their newer chemistry.
So if it is less than that, then we have to repeat again.
Something's gone wrong. Something has gone wrong. It is most likely sequencing process went wrong. Your library was not good or something of the sort.
So library, so obviously we do the quality check.
How you will know? That thing may be good, but if you have used like any ancient DNA, it's not going to be of good quality. So there are many steps involved in this. So generally library prep process was good, but if the sample quality itself is horrendous, you're not going to get good results. So there are ways to offshoot it. You can do UMI. You have to do many X coverage. So that will help remove some of these errors.
Okay. So next up is FASTA file. This is the most basic file. It just has two sections, the header section and the sequence section. It doesn't have, you can say, it doesn't have all those informations that were there in FASTQ header because it did not come from any sequence or anything. It just used to store the sequence information. That's it. We generally use it to store human genomes. The human genomes are generally stored in FASTA files. So you can see out of all these, you can see that FASTA file .fasta, it is human genome. But what are all other files? So those files are basically index files for that genome, and those are required by particular aligners. Suppose we are using BWMM, it is one of the aligners. So it requires index files to align our reads to that genome. So basically it looks up the index, and then it is basically it allows the alignment algorithm to quickly locate the query sequence in the reference genome without having to search the entire genome. So it is fast that way. That's why we require all these index files, and they can be generated by only one command, but it generally takes a lot of time to generate all those indexes. So we can just generate those indexes once, and we can keep it in some folder, and we can use them every time. We don't have to do indexing every time.
The FASTA file we load to the machine. It's the reference files, like the reference JPEG.
Yes, it's a reference. We don't load. Yeah, those are just sequence storage files.
What? This is your normal reference for human genome, right?
Human genome. Yes, so the next step is SAM file. When we align this, align our reads with the human genome, we generally get a SAM file. It is a sequence alignment map, and it has two sections again. It has header section and alignment section. So header section has this SN and LN. SN is reference sequence name. So SN here is chromosome 1, chromosome 2. All those are reference sequence names, and LN is reference sequence length. So chromosome 1 is that base pair, that big of base pairs. So it is general information only. And then that RG, that is RG ID, it's read group. So we basically specify those read groups while aligning to that BWA command. So we can do it what we want, we can specify. So here we have specified like ID as AML, platform is Illumina, then library, we can specify any type of library. So here it was specified library MIPS. SM is the sample name. So this read group is required if you're processing multiple samples together, because if you don't have read groups, then the SAM files won't be, you won't be able to differentiate between different SAM files. That's why you need read groups. And then that CL is just command, which command you use to align your reads to the reference genome. This thing, BWMM, this command we use to align our reads to the reference genome. So this is the second section of SAM file. It is the alignment section, and here you can see it is a very big file. You cannot open these files in Excel and anything. So that's why you need Linux to open these files. So these files are basically here. The first thing is Q name. It's the query template name. You can see, you have seen this in FASTQ only. FASTQ header has the same thing because it came directly from that. So it is basically the read name only. Then this is SAM flag. It indicates the alignment information. I'll go in detail about SAM flags in later slides. So this is SAM flag, just remember as it is. This is our name, so this is reference sequence name. So this read got aligned to our chromosome 9 at this position. And this is the mapping quality. And this is CIGAR, so concise idiosyncratic gapped alignment report. I'll also go over these in next slides. Flag and CIGAR I'll explain in detail. This is just the information about the read, how it is aligned. Then the next is RNext. It's a reference sequence name of the mate. Means like if this is R1, then its mate will be R2. Then it is RNext will tell you where the R2 is aligned. But here it's not given. RNext PNext is the position of the mate. Here it's not mentioned because this is actually a pair assembled file. That's why it means that we assembled R1 and R2 like this, and that's why the aligner thinks it's just single end, so it's not giving us the info of the pair ends. So but if you want to see paired end files here, you can see BAM file is just binary alignment map, and it is similar as SAM file. It is just compressed file. So it is very less size. It is of very less size than SAM file, but it is of the same structure. So here is this file was not pair assembled. So we aligned both R1 and R2 together to genomes. So here you can see this is the SAM flag. This read was aligned to chromosome 1. This is the position. This is the mapping quality. This is CIGAR. I'll go over these. Wait. This is the R name of the RNext. Basically, RNext means the reference sequence name of the mate. So this read has a mate, no? So it got aligned to this position. And this is template length observed template length means it is the difference between R1 and R2. It is the distance between R1 and R2. It is minus 141. It is in negative because its mate is upstream of this read. That's why it's minus 141. Because if this read is here, its mate will be here. So that's why it's negative. And that is the sequence which got aligned. So yeah, SAM flags. SAM flags are basically bitwise flags to describe the read. So if one number is there, so the read is paired in sequencing no matter whether it is mapped in pair. The second is like it's basically the information about the read. If the mate is mapped, then it will say 8. If the strand of the query is strand of the mate, it will say 20. But what happens, it will add all those things, because only one thing won't be possible for all reads. No, there are multiple things. So basically here, see, 147 is our SAM flag. There is a great tool designed by Broad Institute. We can input our SAM flag here and it will say which what type of read it is. So here we input this SAM flag 147. It is saying that read is paired, means R1 and R2 are present. The read mapped in proper pair, means both of the reads got mapped. Read is reverse strand. So this is basically the R2 read, because it is it is mapped on reverse strand, and this is second in pair. So it is the R2 read. So here and here you can see it was different. It was 0. So if you had put 0 here, you would have seen what was that read. So cigar is concise idiosyncratic gapped alignment report. Cigar is basically how our read is aligning to the reference. So M indicates the match, like our the base of our read is matched to this reference sequence. D is deletion, I is insertion, S is soft clipping, and H is hard clipping. I'll explain what those are, but I'll explain cigar first. Yeah, see, this is our reference, and suppose this is our read. We map it like this. This is our read. So it started at position 5. So pause will be 5. Then A is mapped, C is mapped, T is mapped. But then there is one deletion, you can see. Sorry, it's insertion. So you can see here, 3 is matched, 1 is insertion. This A is insertion. Then again you can see 3 are matched. Then 1 is deletion. Then again you can see 5 are matched. So cigar is basically telling us how our read is, how our reads are mapping to the reference. Okay, that was SAM files. So next up is VCF file. After we get our SAM file, we convert them to BAM files, and then we put those BAM files in variant callers, different variant callers. So to basically get the variants. So VCF file, this is the header of VCF file. This is the first section. It has 3 important information. This is the filters, this is format, and this is info. So format has allele frequency, like depth, GT genotype. There are different informations available. I'll tell you about those. I have made a table. This is basically header, and this is the main structure of our VCF file. These are the main 8 columns, like chromosome, position, at which position the variant is present, ID. If that variant is already in our DBSNP database, so it will automatically give you the RSID, which variant is that. Then reference base. So what base was present at the reference, and alternate is what base at this position was present in our read. Then quality is the quality of that variant. And then these are variant caller specific information. So this variant call VCF file was generated by Mutect. So these information is like Mutect specific. You can go and read about that in Mutect documentation. But these are the main fields of VCF. And then info section, we have GT. GT is basically genotype. So yeah, what is genotype? So if genotype, here you can see genotype is 0 and 1. 0 is our reference base and 1 is alternate base. So we have 2 alleles, no? So here it's saying that 0 means one of the allele has reference base and the another allele has alternate base. If it had 1 1, that means that both of the alleles have alternate base, like that. These are the info tables. You can check the VCF documentation. It has like a lot of info. You can just take in whatever is required and ignore the rest. And this is our last type of file. This is bed file. This is basically typically used to specify the regions of interest. So for analyzing the genomic data. So here you can see this is the chromosome, this is the start, this is the end. So we are specifying this region, the region between these coordinates. And these are just comments, but we can name them anything. But this we named like after exons. So this region is basically the exon 4 of GATA2. And this is the NMID. So why do we use these bed files? So these can be used to calculate like coverage. If you're targeting specific regions and you want to calculate coverage in that region only, you give there is a tool called bed tools. You give these bed file to the bed tools and you calculate coverage in that particular region. If you're variant calling and you want your variants inside these particular exons only, then you provide these bed files to variant callers and it will give you the variants in that position only. And this is basically strand, if it is on minus strand or plus strand.
So the bed file we generate during the panel design.
Yes.
Yes. And then we have to feed it.
Yes. That's it. Any questions?
So generally we have questions and discussion now, right?
Okay.
Oh, Arpit talk is now? Okay, sorry. So any questions, please. Sorry, I interrupted.
Can I ask you a question? Like now, with respect to the panel designing, once you from the database you select the genes as Nikhil sir said, and then you generate a bed file with the start coordinate and the end coordinate. Now you told me like by uploading it you will get the coverage.
Yes.
So in which particular software or something?
There is a tool called bed tools. You provide your bed file and you provide your data, whatever you have generated, and it will give you the coverage in that section.
Okay. So what it essentially does, it's not just bed tools. You can use
anything,
bed tools. You can use SAM tools. You can use any pile-up file. You can use GATK's depth of coverage. So essentially, it will tell you for this region how many times it has been read. That's all that it is telling you. It is telling you look at a segment of the genome and find out how many times this has been covered. That's about it. Okay.
So I have a question for the audience. So what is the nucleotide sequence that you get from Sanger sequencing called? So once you load them, or if you put, what do you call that format as? FASTA, right? So what is FASTQ is? With the quality. So FASTQ. So the sequence along with the quality.
Thank you, Vaibhav, for your informative session. I hope it helped fill in the gaps you may have had regarding NGS file formats. Yesterday, we covered sessions on long-read sequencing and 16S sequencing using Oxford Nanopore. To build on that knowledge, I invite Mr. Arpit Mathur to the stage, who will provide us with an overview of 16S sequencing and metagenomics analysis.
Okay, am I audible for? So, good morning everyone. So my name is Arpit and I'm a computational biologist by training and I work at Dr. Nikhil Patkar's lab. So my talk is on genomic tools in clinical diagnostics and why computational biology, bioinformatics is leading the way in diagnosing cases where routinely old textbook, medical textbook fails. So this is an overview of what actually I am involved in. So we are like involved in a lot of sequencing from stool samples, from like cell-free DNA, relating it, benchmarking it with blood culture reports, doing wet labs, gut microbiota studies, and basically decoding crosstalks between bacteria and immune profiles. So my talk will be on two major themes. One will be 16S, and then another is full whole genome sequencing. So like previously told by Dr. Gaurav yesterday that and Rahul, that 16S by nanopore gives a major advantage, and I will convince you at the end of the talk why. And on the other hand, full whole genome sequencing does have some advantages, but the theme of the talk is understanding this biology and medicine via the lens of data, and why it is important. And all through the talk will be genomics, data science, microbiology, and oncology. And lastly, the lessons that we learned from our experiences. Our whole team learned from the last 10 months, and we will try to tell you what you as clinicians can learn from our experiences and help patients' lives. So some historical background, and Professor David published this landmark paper in 1960. First gave those 16S sequences in Bacillus species. Afterwards, in 1987, Carl Woese, he's an American microbiologist, very, very famous, and he describes 16S sequence as molecular chronometer. So molecular chronometer means, it has some sequences which can tell you what the species is. With time, those sequences evolve, and as time evolves, species evolve, and those sequences can tell you exactly what the species is. And this is the latest 3D structure from AlphaFold. The 16S sequences are represented in green, so it's a PDB structure. Structural biology is a different field altogether, but you can see how the construct of 16S is. So 16S sequencing and why it was previously covered by Rahul, but I will give you some insights more. So 16S is a beautiful biological construct that is molecular people love it because it's designed in a way to sequence it. And the sequences you can do like V1, V2, or you can target anything, but why to target full length is a question. So you can see in this graph that all those regions like V1, V2, V3 have some more entropy. So entropy means richness. So there is richness of nucleotide sequence, and you can see some species always have some differences in hypervariable regions. So if you know those hypervariable regions, you know a species. So that's the catch. And if you look at 16S, so in the same paper by Johnson, you can see that V1 to V9 gives you a better coverage of species. And also suppose you have E. coli strain 1 and E. coli strain 2. By V1 to V9 sequencing, you can differentiate between those strains and species. So it is advantageous in a situation where the patient is going through a graft versus host disease, or patients are dying of sepsis. You need to know exactly what the species is causing, and suppose there is an outbreak, so you need to have a specification what the outbreak is. And the V4 regions, you can see some inferences that it is highly unreliable, very less mismatches. So you don't target V4, you target V1 to V9. So that's the lesson, you target V1 to V9 to get a better coverage. Now there are some challenges in the wet lab. There's some target region bias in 16S. Suppose you target V1 to V9, you will get less of protobacteria and like that. So these are challenges. So that's why V1 to V9 gives you a better understanding of the data. Primer bias, so you design different primers, you will lose some of kind of species. And there's a PCR bias, so annealing temperature, PCR cycles, all that also impacts. So these are wet lab challenges, and we face them are facing them during our work here at ACTREC also. Now, which sequencing technology for 16S and why? So there is always a question what to use. So answer is it depends on your aim. It depends on resources you have, the people you have, and so some points like error rate. So Illumina will give you less error rate. Nanopore will give you high. As talked by Dr. Gaurav yesterday. Depth is high in Illumina. Nanopore is low, but with good like Promethean grid ion, you will increase that. Cost is high of Illumina. You require a proper setup. Dr. Shweta and Dr. Swapnali told yesterday, you need a proper AC room with no dust. Nanopore doesn't have that, problems, but you need some hygiene around. Turnaround time, Nanopore is very fast. Resolution, now this is important. Resolution is species level, so you need those details. If you have an antimicrobial report by Nanopore sequencing, it is more reliable. Now the important point, as a computational biologist, I will always try to say that Illumina is for now the king, because the downstream analysis that you can do by Illumina data are like very high. You can do a lot of functional analysis, lot of things. Nanopore is building up, but the data, the algorithms are not built for long read as much as Illumina, but with time they will equalize. And last point, this polymorphism genome. So Illumina will not able to identify those polymorphism, but Nanopore can do. And just to give you an example, that in Haiti, so Haiti is a country in between North and South America. There was an outbreak of chlorella in 2010, where 10,000 people died and 8 lakh cases. And why 16S is important? Because this outbreak was, identified by whole genome sequencing of 16S. So you can, see the, why 16 is important. It can, save a lot of lives. So I will go through the databases that are used for 16S profiles. Now the FASTA reference sequence that Vaibhav just told today. So FASTA is just a reference, while the index databases. So index is just a like a college and a school libraries, where every book is sectioned in a particular way. So it makes your algorithms much faster. You know exactly where to go. So that's why indexing it does. You have a coded databases, so you know where to go, which contigs and scaffolds to look for. So particular indexes like BOTI2 we used in clinical diagnostic also. Kraken and Bracken, which is a advanced version of it, we particularly use in metagenomics. BWA, that is BOTI Wheeler algorithm, so it is again used in clinical diagnostic also, and also in metagenomics. And Kaiju is a protein based database. So lesson is index databases are more computationally feasible, doable, because they are fast, they are coded. Okay, now algorithms. So Dr. Vishram told you about pipelines, but the idea is in data science, in computational biology, the heart is algorithms. Pipelines are assemblies of algorithms, assemblies of doing different tasks. It's a management system, but the heart lies in what the algorithms is. So for short read data, you can use these in red. We use Kraken, Minimap. So every algorithm is different in a way. So it's a completely different field in computer science. And just to give you an example, BOTI2 was developed by Johns Hopkins University Department of Computer Science, and it is used in every clinical lab, in genomics lab, to align your sample with the reference human genome, maybe HG19 or 38, whatever you use. In long read, Minimap 2 works. So that's why long read does have low algorithms that are developed, but with time it is gaining importance. People in computer science are now trying to find more algorithms. But the heart is those machine learning and statistical methods, expectation maximization algorithms that we use here at ACTREC, Bayesian inferences, hidden Markov models. Just to let you know what is hidden Markov model, so there was one question how base calling actually is performed. Okay, so in nanopore, that is basically it's a signal processing technique. The electrical signals that are produced by, when the DNA goes, and the electrical noise that Dr. Gaurav was talking about. So there is a hidden Markov model, deep learning model, which is pre-trained to filter out noise from that signal. So it's a signal processing technology. So hidden Markov model is used in that. So it is called Dorado, the algorithm used in nanopore. So it is highly efficient, and there is a new in the market called Hero. Its name is Hero. So it gets you more accuracy. So with time, accuracy of nanopore will be in contrast to Illumina. So that is the way. Okay. So general workflow, how this 16S goes, Rahul has talked about it, but simply like you take the stool sample from the lab, from the patients, and you extract the DNL by different methods, you quantify it and build the library. And then you can do nanopore sequencing, and so base calling algorithms like I talked about. We use Dorado as of now. You get the FASTQ file and you do the taxonomic profiling. So I will go about how taxonomic profiling is built, but the idea is you have the patient 16S data. You remove the negative template controls to remove the wet lab errors that we have, and also the biologically negative controls, which are actually the healthy people. So you remove those background noises and to tell exactly that the bacteria that is causing the problem actually rises up in the table. Then you can do a quality control, and then you can do downstream analysis. So one example of the pipeline that we use is called EMU. EMU is a species in some part of the world. But like I told, the heart of pipelines are the algorithms. So it uses those expectation maxima algorithms. So I will not jargon about it, but one word I could say that it iterates. This algorithm is an iteration algorithm till you get the best alignment the read can get it. So it is the the best in the market as of now. Lot of pipelines and use it in cell-free DNA. We use it in circulating tumor detection. We use it. Okay, so you get an taxonomic alignment profile like this. So this patient gets an abundance table like that. So Escherichia coli 13% abundance, and likewise. Now you can do downstream analysis. So once you get the abundance table, you can use ecological indexes to quantify your taxonomical profile, and I will show you how the clinical importance it is. You can do functional analysis. So this paper by Nature Biotech tells you which pathway are enriched in your stool sample. So a lot of physiology and biochemistry, you can gain those insights about. So this is how the tables look like. So from the stool sample you can see alcohol dehydrogenase pathways enriched, and likewise you can also have pathways like glycolysis, TCA cycle. So you can gain those insights, those biochemistry insights from simply a nucleotide FASTQ data. So like I told, I will try to convince you why 16S is important. So this is data from our own clinical trial, where patients who have gone through bone marrow transplant, their microbiota profile goes very down. And there is a good concept, established concept that gut microbiota changes do impact survival of patients during bone marrow transplant. So this is how 16S is important to real-time monitoring of patients, and maybe give them probiotics, prebiotics, or some kind of therapy to improve that gut microbiota. And if you do this fecal microbiota transplant, it increases. So it's a good practice to do fecal microbiota maybe before BMT, who are patients doing worse. So it's a clinical importance thing. And you can use quantify wellness index, or basically say how much good bacteria is and how much bad of this. So that is possible. You know what is dysbiotic and eubiotic profile. So it's simple, matching of data. But there are drawbacks. 16 of the data, it may be a 16 long read or short read. Simply you can't do a good resistome profiling of 16. Antimicrobial threat antibiotic resistant threat that we are all going through. It will hit us in some days, in future it will definitely hit us. So you can't build those resistant profiles simply because the genes are not present in the 16S data. You can't do proteomics, you can't do lot of downstream analysis from 16S. Only few, but not more. So that's why whole genome sequencing is important. It may be any source, maybe plasma, stool, sputum, or even now there are things called skin microbiome. So the workflow of library is almost same, not exactly 100%, but for every sample you centrifuge it, and by different extraction method, some use lysis, some do not. You do cleanup, elution before you do quality check, and then prepare the library according to the Illumina nanopore device you have, and then do a bioinformatic analysis. Now this is important. So even if you do not love microbiology, this concept is very important to understand how data is actually in genomics. So even if you do like cell-free DNA or circulating DNA, liquid biopsies growing up, you will always have a noise in majority and a signal-isness. So I am an engineer. I know we have to read a lot of books called signal to noise ratio books. So it essentially tells you how much signal do you have compared to noise. And in metagenomics, the noise is your human genome actually. So you have to remove it. Now how to remove it computationally, I will tell you. So these FASTQ files just shown by Vaibhav, you can see different reads. So you have to identify the human genome reads and just mask them or remove them. I will prefer masking them. So when you remove the human genome, you have a good concatenated, targeted bacterial or a contam, and then you can do a lot of. And why it is important is because if you don't remove the noise, the patient spectrum remains constant. Suppose, and that's the problem that we faced some days back when we sequenced around 100 patients of data through Illumina. We just put these data in the pipelines. Every patient has Neisseria gonorrhoeae on the top, which is not clinically relevant. So we thought what went wrong, and we forgot to remove this human genome. So we do masking of human genome per se, because masking means you basically change those ATCG with N. So if there is a FASTQ file and if you see an N in the sequence, it means the machine will skip it. It will just count it but skip it, will not align it to anything. So that's another lesson. You remove and you see the spectrum of patients different. So that's not the idea. Okay. Now how to do that? So trimming is one of in workshop we have trimming by Trigonomatic. You can use different tools. I use FastP. So basically this is the command line. You have to trim all those repeated sequences and all those Illumina adapters. So and the minus Q is quality score 20. So in metagenomics we keep quality score 20. For clinical diagnostic we take it to 30 or 38 something. So basically you mask them and not remove them. That's my suggestion, but you can always do experiments about it. Now the concept of abundance. So this is very much important. Every algorithm per se will give you reads aligned to a particular species. That is fine. But can you by confidence say that these reads are measure of abundance? No, you can't. because like Klebsiella pneumonia, here you can see have 1000 reads, but its abundance is not the highest, it is the lowest actually, because in metagenomics there is concept called depth of coverage, where you divide those reads by the genome size and get those abundance values. So that is important. So it is similar to what clinical pipelines, our clinical fellows go, they also see for coverage. We are also doing the same, but we are including breadth of in depth both. So this is not the way. Now, like I said, antimicrobial resistance can't be done by 16S, but it can be done by whole genome sequencing. And we know this antimicrobial threat, it will almost kill 10 million people by 2050. So detection by detection by whole genome sequencing, we can detect those AMR genes, we can build those resistant profiles, we can detect an outbreak very early, and get to a conclusion very fast. Some databases, anybody who works in public health knows this database. This is called CARD database by Canada. It is a regularly updated database, even better than WHO. Almost a week there is an update of which species has what kind of strains, and lab around the world who are working in bacterial genomics updates those databases regularly, so you know which antibiotic is prominent. There are more by Denmark, like ResFinder, Colorado State University, and by NIH also, but like I said, CARD database is the top choice for most people. You can do functional profiling. Functional profiling means proteomics, metabolomics, and why it is important. So how those bacteria cause problem, pathogenic bacteria, basically they secrete metabolites, right? So you need to profile those metabolites, you need to quantify how much metabolite is secreted, and then you can decide the treatment regimen of the patient. Right, okay. So you have the metagenomic FASTQ data, you can just use this pipeline by human too, and you can get proteins. So FASTQ data, nucleated sequence into proteins, there is a system called PROKA, PROKK. You can do that. You can do metabolites. Once you have got proteins, you can do metabolite secretions. So you get what lactate or butyrate abundance is in the patient's gut or in plasma or skin, whatever sources. You can do bacterial network analysis, pre-treatment, post-treatment. And why it is important? Simply because, so I will mark this something on 24th September where our workshop actually started. It came just at the time where you can see those bacterial community have behaviors according to the friends they have. So this is important. We need those bacterial associations and dissociation networks. Lastly, I will leave you one thought before the lunch. We all know who works in cancer of this paper called Hallmarks of Cancer. We all know Professor Hannan and Weinberg professor. So they gave you some 10 odds reasons why a normal cell becomes a tumorigenic or metastatic or neoplastic. But in 2022, after 11 years, then the gut microbiota was included in that. So that's established norm now to profile microbiota. And whatever the source of microbiota is, it is affecting people, it is affecting health, and in hematological malignancies it is also important because it changes and hijacks those immune profiles, especially those lymphoid lineages. Lastly, before you go for lunch, you make sure you have it, and if it is there in the menu. Different diets have different metabolite profiling, and this is a star marked diet. So it came yesterday, this gut microbiome. So now gut microbiome is the new trend in the market. Even if you don't want to go through in metagenomics, but principle of genomics you will learn a lot from metagenomics. So that's my summation at the end. So thank you for your time, and at the end I will like to thank Dr. Nikhil, Dr. Anand, and everybody at faculties, clinical fellows, Dr. Gaurav, Dr. Shweta, Swapnali, and Shruti ma'am, because this research field is highly interdisciplinary, and you need a lot of collaborations, talk with people, and some talks, conversations can upgrade your research profile a lot. So with that, I end my talk. If you have any questions, please.
So one comment before anybody asks question. Arpit, very nice talk. Arpit is a very bright young man. But before anybody asks, can you tell them what, or maybe audience can tell, what is use of 16S sequencing? Anybody knows about this can say in his own words. क्या है क्या ये कौन सा चिड़िया है? All of you have done MBBS, I think within 7-8 years of me, maybe 10 years of me. That time this was not there. So who can tell? So when we read microbiology, how did we know what is causative organism? What you used to do? Culture, right? So where do you think 16S comes into this? 16S is a revolutionary technique. It is still unfortunately not at all common in India. But in the West, this is like the first thing to do. If you see a pus, if you see anything that is there, infection, it can be whatever lesion, whatever secretion is there, you first do a 16S. You send for culture, but you do a 16S, because this is culture-free method of identifying pathogenic organisms, and that is why it is so fast. Many of the organisms can't even be cultured. All right, they don't grow under culture. If you give antibiotics, those bacteria go away, but 16S stays. The second thing he described was metagenomics, where in 16S is like targeted sequencing. You're targeting something in bacteria. This the second cell-free DNA that he described is known as shotgun metagenomics or whole genome metagenomics. If we know from COVID era, this is how we identified the COVID bacteria, by doing metatranscriptomics, because it identifies RNA viruses as well. So metagenomics or DNA based will identify anything in DNA. All right. So that is what he explained the computational pipelines for. Now you can ask him technical question. They don't know. Most 99% people are not aware of what is this use or why you are giving this talk.
Right. Okay.
Arpit, please answer.
So if you go for nanopore, it is quite cheap. Now flongals have problems, but if it works, it's quite easy.
So flongal is extremely affordable. Flongal costs 7,500 to 8,000 rupees. On one flongal, you can run 3 16S samples. 16S costs approximately 900 rupees or some sort for library prep. So it is negligible to do flongal based work. But as he said, flongals are technologically not up there yet. It's a bit of a hit and miss is what we are seeing. In our lab, you still require have molecular lab, you require extraction, controls, computational, this thing, but the Anand sir is there, he is running a clinical trial on this. Maybe Anand, few lines on what you are doing you can tell.
So there is an NEJM paper in 2022 that was by Memorial Sloan Kettering team. So MSK has done a lot of work in microbiome in BMT, where they found that the day post-transplant, the gut microbial diversity on day 14 is an important predictor of mortality, because day 14 is around the time when the stem cells engraft, the T cells, the nascent donor T cells which have engrafted are in circulation, and that time if the gut microbiome is bad, the bad gut microbiome cells signals to these T cells to be attracted to the gut. And the T cells, if they are attracted to gut, then they get exposed to the alloantigens, and that kind of triggers the GVHD. The same thing if the gut microbiome on day 14 during peri-engraftment period, if the day 14 gut microbiome is good, then the T cell drawing to the gut is lesser, and that is supposed to decrease the chances of GVHD and hence reduce the transplant related mortality. So in about more than 800 patient cohort sample in a multi-centric study, this was what was shown by the MSK-CC team. So now that there is some data to suggest that the microbiome may have a role, so we are trying to see in our cohort whether modulating the gut microbiome actually helps. So we are using a kind of a drug to, in a randomized manner, to see whether the gut microbiome is different and whether that impacts outcomes. So this is still under research, and it's a long time till it comes into a clinical. So that is during the conditioning, just before the end conditioning to post engraftment. Yeah.
So what drug are you using to modulate
we are using rifaximin. So there is German data. So rifaximin is one of the drugs that has been used as a antibiotic prophylaxis. So metronidazole, levofloxacin, rifaximin. The different centers have used different drugs. So there is no level 1 evidence to suggest that one drug is better than the other, or to suggest that even microbial prophylaxis is actually helpful. There is no level 1 evidence. But in our setup, mortality due to infections and GVHD is a major hassle and cause of concern for us. So we are trying to see whether modulating the gut actually helps us. It's still a clinical trial. We don't know how it will turn up.
You also mentioned something very provocative about fecal microbial transplant or something like that. So I think that has probably been abandoned in few trials because there have been patient deaths. Like 2 patients died in a FMT trial because they got a resistant E. coli from the same donor. So I think just to suggest that FMT might be useful, I think it's with a pinch of salt.
So I'll take that question. So FMT is an FDA approved modality of treatment of Clostridium difficile diarrhea. So if somebody has Clostridium difficile diarrhea which is not responding to your standard vancomycin and standard drugs, then FMT is an FDA approved method of clearing the Clostridium difficile diarrhea. So it has been used in C. diff. For transplant, it is being explored in steroid refractory gut GVHD. So there.
Patient himself or herself would be immunosuppressed, and then you are giving a fecal transplant which might have potential pathogens.
So that's what I said. So I am not saying that it is the modality of treatment. It is still under clinical trial. There is a phase 1 data to suggest that there is safety, but the efficacy is not yet established. So it is still not a proven therapy in BMT. It is still under clinical trial. So people have used it even in India in basically in completely refractory cases as a kind of a salvage or as a compassionate basis because there was nothing more to offer to the patient. But it is still in clinical trial, so it is not a standard therapy, as you are correctly saying. But it is not that it's not used. So for some indications, it is approved. So FMT is also being used in inflammatory bowel diseases, refractory
UC and
So, but there also there is no level 1 evidence to suggest that it works. The only place where it is FDA approved is C. diff.
Which is refractory. Okay. And coming to the mechanistics, how you're saying that the T cells are being home to the gut and all. So are these microbes, they are secreting some chemokines, or is there any pathogenic mechanisms involved? If the microbiome is changed.
That is not clearly known.
Okay.
And still, we are also trying to explore that. So
whenever the study you've designed is just exploring all this.
Whenever a patient in our study will have GVHD, and if in case we do a gut biopsy, so we'll be seeing.
So you're doing an immunohistochemistry also for the kind of T cells which are infiltrating there.
We are trying to sequence the gut. We don't know how exactly we'll do it, but we will try to see how it's an exploratory endpoint in our study. But the
sequencing is a single cell, or is it like a bulk
So it's a single cell. I mean, I'm not exactly.
क्या बोला?
He said that
tissue.
for the gut biopsy, he's going to sequence the gut biopsies. So is it a single cell?
Single cell.
Single cell.
Single cell. So we are going to take the biopsy and then we are going to make it into a single cell suspension and then see what is the crosstalk between the epithelial cells and reaction and the epithelial cell and all the milieu.
So the theoretical explanation to this is that there are certain toll-like receptors on the surface of the bacteria. And there are also certain butyrates and certain metabolites that are secreted on the bacteria. So the toll-like receptors are the one that come in contact with the gut epithelium, especially in the crypts of the gut epithelium. There are certain Paneth cells, and the toll-like receptors can be dissolved, and they can cross through the mucosa and come in contact with the T cells, which are in the submucosal layer. So that is how it is supposed to impact the local immunity. How it impacts the systemic immunity, we don't know. Yes, that's how we are doing it. Yeah, we are doing it at baseline and then subsequently during post-transplant.
I have a question. Shall I? You talked about antimicrobial resistance.
Yeah.
We can detect with the help of that 16S sequence.
Not by 16S, but by whole genome.
By whole genome,
Yeah.
right? So we also have in bacteria plasmids, which are independent and may not integrate into the genome. So they also carry some genes which are responsible for the resistance. So do this technology will include plasmid?
It's a whole genome, so whole genome will include plasmid also. So you can classify plasmid according to the different alignment. Then you can do downstream of that. So it is possible.
So when we say whole genome, these are actually what we are doing is sequencing cell-free DNA of patient. Because even though there is sepsis, there are macrophages and other activity active throughout the body, which is biting it, which is taking the genome of the bacteria, digesting it and spitting it out. And that what gets spit out everywhere is cell-free DNA, and we are actually catching these fragments of bacterial DNA or something and sequencing them and trying to find out what it is. So yes, it can detect AMR as well. We are working on it, but it requires, all of this is frightfully expensive and it costs a lot of money. So especially for sequencing. So that's the drawback of this.
Okay, thank you.
So we have 12 and 11:30 to 12. 11:30 to 11:45 we have break. So we have two options. One, we can have question answer discussion session, or we can hang out and come back at 11:45. What would you prefer? Who is that fellow?
We can take a short tea break of 15 minutes and come.
चलेगा, वैसे करो. So we will take a tea break and maybe hopefully tea has started there, but you can just generally break and come back. And anybody who wants to ask questions can come, feel ask me, Dr. Shweta, Dr. Arpit, Dr. Vishram, whoever. Vaibhav.
Like for the